Greetings, this is chapter 3. This is the last section, section D, and this is going to be about cell cycle. It's going to be about mitosis. It's going to be about DNA replication, RNA transcription, and protein translation. So away we go. Not every cell in the body, but many cells of the body, about half the cells in the body, will undergo cell division on a daily basis. And that means that they're going to cycle from cell division to active expression, and then they will have to divide again and then have a period of active expression. And this is called cell cycle. Cells will cycle between division and expression. And in the olden days, back at the turn of the 20th century, you just had histologists. So the electron microscope was not invented yet, and biochemistry was really not a science. And that meant that looking at a cell under a microscope, it seemed as though you had a phase between cell divisions, which was called interphase. And then the cell would divide by mitosis. Along comes biochemistry and molecular biology and cellular biology, and now we start to realize that interphase is quite complicated, and it's going to include 3 subphases, G1, S, and G2, which I'll explain in the next slide. And the old terminology, though, has stuck. So you could be talking to a person who is talking about a cell interphase, and that's probably a histologist. And then you could be talking to a person who is a biochemist, and they would be talking about G1, S, or G2. And then cell division, mitosis, is something that even the histologists understood the different phases because they could see them. You cannot look at a cell and go, oh, hey, that's in G1 under a microscope. It's one thing if you have a lot of dyes and you have a scanning electron microscope and you have all the bells and whistles of the later 20th century and the 21st century, but prior to that, the histologists really just thought there were 2 stages, interphase and mitosis, and now we realize it's a little bit more complicated. So we will explore some of the complications. So interphase. What I would like to do is show you this picture. I tend to be more of a picture person. So this is the cell cycle. You have a cell, and it's going to divide into 2 new cells. Here they are, cell 1, cell 2, from the initial cell. And in a bit, we'll talk about the different phases of cell division. We'll talk about prophase, metaphase, anaphase, telophase, cytokinesis. Now, once the cell has divided, and now you have 2 copies, they clone themselves basically, you have the G1 phase for growth. So it's going to grow, it's going to express itself. If this is a cell that is going to make digestive enzymes, then that's the time it makes the digestive enzymes in the G1 phase. Now, knowing that it's on this clock and that it has to replicate itself, make a copy of itself, it will eventually swing into the next phase, which is called S. It stands for synthesis, but what really is going on in S is that the DNA, the chromosomes, the genes are going to copy themselves. DNA replication occurs, and that has to happen right before the cell divides so that each cell will have a full complement of chromosomes. We have 46. So in this S phase, that 46 chromosomes are going to duplicate themselves, and now you actually have 92 chromosomes. Now you don't have that for long. You're going to go into the G2 phase, which only lasts for a couple of hours. And that's a phase, yes, the G stands for growth, but it's going to make whatever it needs to make before it divides. And what I mean by that is you want both new cells to have equal amounts of organelles. So you might have to make another lysosome. You might have to make another mitochondria. And that's what you're going to do in the G2 phase, just to make sure that everything is now set for cell division. And then you're going to get into mitosis and then go through these phases and have the cell divide, and this will then continue. Then it will go into G1 again, replicate its DNA, and thus finish any organelles that need to be synthesized in G2, and then you're going to divide again. So that is cell cycle. Now there are some cells that leave cell cycle and they go into what is called the G0 phase. And those are cells that are so specialized, so complicated, so differentiated that you never can make them again. So if someone has a heart attack and they lose a lot of myocardial tissue in their left ventricle, they are never going to get that back. And they are going to have a certain deficit in their cardiac function. If a person has a stroke and they lose a lot of tissue in their brain, the brain can rewire up to a point, but they're never going to get those neurons back. So there are certain very specialized cells that don't do cell cycle anymore. They did when the person was a growing fetus, but it isn't going to happen as an adult. On the other hand, all the other cells, epithelial cells, connective tissue cells, etc., are always doing cell cycle. So this is just showing you, if you're a histologist, this is what you would see. You would just see loose strands of chromatin, nucleolus, the nucleus. You would see maybe some of the organelles, depending on your microscope. And here you go with some centrosomes, sorry about that, centrosomes, but you're just not going to be able to say, oh, that's G1, hey, look at this, hey, there's G2. That you can see on a biochemical level, or if you have an electron microscope, but you're not going to see it with a simple compound microscope that they have you look at cells in lab. But they do occur. Okay, now this is going to be about that S phase where you're going to make copies of your chromosomes. It's called DNA replication. That famous double helix where you have your A's and your T's and your G's and your C's. And that codes for all the blueprints for making everything that a cell needs to make. So obviously if you're going to divide in a few short hours, you're going to want to make two copies. One copy is going to go in one cell, one's copy going to go in the other cell. And this is the process of DNA replication. So remember that in that S phase, you double the number, 46 to 92, and then in mitosis, you're going to make two cells. One cell is going to receive one set of 46, the other set of 46. And this is the phases of DNA replication, which I would like you to know. It's that middle part of the molecule where the A's and the T's and the G's and the C's, the nucleotides, are held together by very weak hydrogen bonds. So the weakest link is the hydrogen bond, not the covalent bonds, which are in the rest of the molecule. And so the weak link breaks the most easily. And so you divide the molecule once it unwinds down the middle, straight down the middle. In the end, you're going to get two identical DNA molecules formed from the original, and this is then you're going to move the molecules in mitosis. It's called semi-conservative replication because you take the original strand and you make a copy of it. So you conserve the original strand and then you make a new copy of that. So one strand is new, one strand is old. And then the base pairing rule, we know that since it's a double-stranded molecule, you're going to have 4 types of nucleotides: adenine, thymine, cytosine, guanine, A and T and C and G. And since it's a double ladder, the A's always bind with the T's, the adenines always bind with thymine. That's part of the base pair rule. And the other base pairing rule is that the cytosine always binds with guanine. And this is showing a kind of color-coded, where your adenine is green, thymine's yellow, cytosine is blue, guanine is red. So you can see the base pairing going on right now. The enzyme that does this, that makes the new copy, is DNA polymerase. I do want you to know that. And what it does is it unwinds that double helix so that new A's and T's and G's and C's can come in and obey the base pairing rule and then make a new DNA strand. So the thing I want you to know is the base pairing rule and that DNA polymerase is the main enzyme involved in making the new strands. Okay, so once we now have our copy, we're now ready to divide the cell into 2 new cells. And that's cell division, mitosis. Every cell in the body does that except the sex cells. And we will cover those in A&P too when we do reproduction, but that's how you make sperm and eggs. Every other cell that divides in the body does mitosis. So it has been broken down by histologists into 4 stages: prophase, metaphase, anaphase, telophase, and then that last stage, telophase, is when the cell actually starts to pinch in the middle and starts to divide the cytoplasm into 2 cells. So that's cytokinesis and the formation of a cleavage furrow, which will then split the cells into 2. All right, so we're going to go through each phase very briefly. In prophase, the nuclear envelope is going to disappear. And the chromosomes go from loose chromatin to very beautifully structured chromosomes. And that happens as you condense the chromosomes, and the histones in the DNA are going to condense until you see these visible chromosomes. The other thing that happens is that the centrioles are going to divide and move to opposite poles. And that's going to become important because they will throw out these beautiful spindle fibers that will connect to a central protein button in the middle of the chromosomes called a centromere. And then they're going to be able to move the chromosomes around. And here's a pretty picture of it. So you can see the membrane is disappearing around the nucleus. The centrioles are dividing. Now one's at the top, one's at the bottom. And then the spindle fibers, these yellow threads, are going to be spun out of the centriole and attached to these. See right here, these they look like yellow buttons or yellow circles, and the spindle fibers attached to them. Now, mind you, the spindle fibers attach to the button that holds the chromosomes together, but they don't touch the DNA because you don't want to damage it. So that's prophase. Metaphase, easy peasy. The spindle fibers kind of tug at the chromosomes so they line up just like little soldiers in the middle of the cell. So here are the middle, the metaphase. They're all lined up and they're ready to proceed into anaphase. What happens in anaphase is that button-like centromere is going to split, and the spindle fibers are going to start pulling the chromosomes towards the opposite poles. And a picture of that is on the next slide. So here is a spindle fiber, and it's pulling it towards the top. This spindle fiber is pulling this towards the bottom. So let's say this is chromosome number 1. A copy goes to the top, a copy goes to the bottom, and the same thing, let's say this is chromosome number 2, one copy goes here, and the other, and so forth and so on. So that you're going to have a full complement. Now, they didn't do your 46 because it would just be such a messy picture, but you're going to have 46 chromosomes go to the top, 46 to the bottom. And anaphase, now the reason why you want to do this, why do you want to do this? Because you're going to divide the cell in the middle. And you want to get the chromosomes at the top and the bottom so none of the DNA gets split, so you don't make, by accident, an extra chromosome in one cell, another cell lacks a chromosome, you don't want to damage the DNA, you want the entire chromosome to reach the top or to reach the bottom. And since the cell always divides in the middle, you want to make sure that there's nothing, no DNA in the middle of that cell. So you pull everything to the top and the bottom. Telophase. Now that means the chromosomes have arrived to the opposite poles. And once you've got that done, now you can start to reform the nucleus as the chromosomes start to unwind into the strands of chromatin once again. And then, as well as the nucleus reappearing, your nucleolus reappears, and the spindles disappear. And at the same time that you're doing this, you can now get that cell to divide in half. And that's called cytokinesis, the division of the cytoplasm. And you always split the cells in the middle and form a cleavage furrow, it's called. But you're going to form new plasma membrane so that when they split, you have two identical cells, 46 chromosomes in each, and you have divided the cytoplasm evenly for the two cells. And that will be the end of mitosis. So this is showing you that the telophase is the reforming of the nuclear membrane. The chromosomes are going to decondense into strands of chromatin, and you're going to reform a nucleolus, and the spindle fibers have disappeared. At the same time, cytokinesis goes on, and this is showing you the cleavage furrow where you're going to make more plasma membrane. And in the end, you've got two cells, identical copies. They've cloned themselves, and everybody lives happily ever after. Then once you've done mitosis, you are now in the G1 phase, and the cell has to make proteins. They have to make enzymes. They have to make structural proteins. They have to be robust and do whatever they're supposed to do as far as helping the body function properly. So it's called protein synthesis. And what you're going to do is you're going to take the gene, the DNA coding for a particular protein. Let's talk about protein X. And 3 of the DNA nucleotides, the A's, the T's, the G's, and the C's, are needed to form one amino acid because amino acids are the building blocks of protein. And there are about 20 different types of amino acids, so you can make all different types of protein. And the code, the genetic code, is actually a series of these 3 nucleotide triplets that represent different amino acids, so that you're going to have that in the DNA. And that is going to be the blueprint, which you're going to have to copy because the chromosomes are not allowed out of the nucleus. So you have to make a copy of the gene. That's going to be your messenger RNA. That can leave, and then the ribosomes will cluster around the message and bring in your amino acids in the proper sequence to make the proper protein X. So that is what DNA is, the blueprint. It's called a gene. You got a triplet codon of your A, T, G's, and C's to represent the different amino acids in the polypeptide. All right, now there are actually 3 types of RNA, and you're going to find messenger RNA, which is going to be the copy of the gene, ribosomal RNA, which we've talked about ribosomes before. They bind to the message and ensure the proper reading of the message of the blueprint. And then transfer RNA is going to transfer the amino acid from the cytoplasm to the proper place on the message that's being held by the ribosome. And then amino acid by amino acid is going to be attached one to another until finally you reach the end of the message and an entire protein is made. And the RNA is a very closely related molecule to DNA, except that it uses uracil instead. So this is the overview, which I'd like you to know that here's your DNA, here's the gene that you want to copy, is the blueprint. You're going to make the messenger RNA. Transcription to transcribe means to copy. So you're going to copy it in that closely related molecule of RNA. In our cells, we're complicated, so we actually can process and kind of cut and splice the RNA to make lots of different types of proteins. So we are going to process it a bit. So you make a pre-mRNA, and then you process it to make the messenger that's going to leave the cell and go out into the cytoplasm. In the cytoplasm are your ribosomes, are your transfer RNAs, are your amino acids. So that's really where you can construct or build. You don't build the protein in the nucleus, you build it in the cytoplasm. There's more room, and so forth and so on. It doesn't seem like there's a lot more room, but there's a ton more room in the cytoplasm than there is in the nucleus. So translation or protein synthesis occurs in the cytoplasm. Here's our ribosome, here's our messenger, and here are the little amino acids which are being pulled together and arranged in order to eventually form a polypeptide, and a large polypeptide becomes a protein. Okay, so protein synthesis is two-step. You have to make a copy. You have to transcribe the DNA into a messenger RNA, and then you take that message, bring it out into the cytoplasm, and that's called translation. The messenger RNA or mRNA is going to be decoded or read to assemble the polypeptides, which will become proteins, and that's done on the ribosomes. All right, the genetic code. I don't want you to memorize the genetic code. I don't really need you to know how to use the genetic code. But the genetic code is a way of understanding which triplet nucleotide sequence stands for which amino acid. So you have different nucleotide triplets for each of the 20 different amino acids. Some of the amino acids they are redundant. In other words, you can use more than one triplet to get the same amino acid. You know, it's like the cell hedges its bets, so to speak, so it has backups. But I just want you to know that the genetic code is a way that we have of understanding which nucleotide triplet or codon represents which amino acid in a protein. So I like this because it kind of shows everything. So here you have your original DNA, here's your messenger RNA, comes out into the cytoplasm, gets bound by a ribosome. And then the transfer RNA picks up the right amino acid, lands on the right codon or triplet sequence, and then you're going to repeat that as the ribosome moves down the message. So you have this ever-growing chain of amino acids that are bound to each other to form a polypeptide that eventually becomes a very large protein. So here it is moving down the messenger RNA, and everybody's getting larger until you've got this really huge polypeptide chain, which would coalesce into an even larger protein. And so there is a stop command showing you the or showing the ribosome that's reached the end, and then the polypeptide chain is released. Now, it could be released into the endoplasmic reticulum, it could be released into the cytoplasm. Okay, so most of the proteins that are made go into the endoplasmic reticulum, specifically the RAFIR, because RAF means the ribosome. So the ribosomes are the place where the protein is made. The protein then enters the endoplasmic reticulum, this tube-like structure that then moves down and eventually gets transported probably to a Golgi apparatus, which will concentrate it and either export it or import it. So here's a summary slide. So when your A and binds with T and your G binds with C, that's called complementary base pairing, and that is going to direct the transfer of the genetic information in the DNA eventually into an amino acid sequence of a protein. So you take your DNA, you make a messenger RNA, and then the message goes out by a ribosome. A transfer or tRNA brings in the amino acid. You move down and read the entire message, and you build up all these amino acids into a chain like beads on a necklace. And then that chain eventually, when you reach the end, becomes a polypeptide, becomes a protein that is released. And there is another slide. This is the last one for this particular part. This is just another way of showing you the same information. So the flow of information is always DNA to messenger RNA. Messenger RNA goes out into the cytoplasm, which interacts with the ribosome and the transfer RNA, and then the transfer RNA brings in the different amino acids, which are linked one to another until you reach the stop, and then you're done. You've made yourself your protein, and the cell lives happily ever after. So that is the end, and I will see you for the next unit. Thank you.